chapter 16 on page 267, Biotechnology and Genomics. On this page, on page 267, on the right side you have the concepts. If you just read through the various concepts that are provided and you understand what the concepts are saying, it will mean that you understand the chapter. More specifically, within the chapter, section 16.1, DNA cloning, you must know what DNA cloning is, and the figure 16.1, cloning a human gene, gives you an idea of how it is done. You should be able to explain how this takes place in a descriptive manner. If you need to draw diagrams, be able to do so, but I will not require you to draw diagrams, but you should understand. An easy way of trying to understand how this is done is to look at the diagrams and draw it out, and you'll figure it out yourself. Recombinant DNA technology, you must know the definition for that. You must know what a plasmid is. I do not exactly need you to know this figure and what the sticky ends are and how the restriction enzyme works, but you must know in general what DNA technology means. For the polymerase chain reaction, the PCR, you must know what the definition is, how it is done, and how do you analyze these DNA segments. What exactly goes into DNA fingerprinting? On page 270, figure 16.3, if it is provided to you, you should be able to identify who the father of the child could be and why. So be prepared to give a small descriptive reasoning for that if you're asked for that. For 16.2, biotechnology products, I just want you to know what is a transgenic bacteria, what is a transgenic plant, and what is a transgenic animal. And for each one of these, the transgenic bacteria, the transgenic plants, and the transgenic animals, you must know one or maybe two examples of each one. Look at the textbook, it gives you sufficient information on that. For cloning transgenic animals, the procedure is laid out in figure 16.5. I will not question you on that. I will not ask you how it is done. Just know at the back of your mind how that is done. Section 16.3 in Genomics, you must know the definition for genomics. Do not worry too much about sequencing the bases, the genomic comparison, the HapMap project. Be able to talk or define the genetic profile, proteomics, bioinformatics. There is a health focus on page 274. Read through that. I will not question you on that, I will not test you on that, but that is good information, and if you've read the chapter properly at this point, you will understand what is being said in this section. For gene therapy, I want you to know the definition for gene therapy, that is what it means, with an example of a disease or two that might be cured or may already be cured or treated by this process. That is pretty much it for this chapter. It's pretty straightforward. The rest of it, it's not a bad idea to read through the summary on page 277. If you read through the summary, it gives you an idea what the chapter is about and gives you a better understanding of the topic itself. If you would like to, you can test yourself. The questions are provided on page 278 and 279, and the answers are provided elsewhere in the textbook. That is it for chapter 16.